Hi, I'm Richard Sever from Open Archive in Cold Spring Harbor. I'm here with John Sedivy from Brown. Welcome.
Thank you.
We're here at the Cold Spring Harbor Symposium on senescence and aging, and you're going to be talking about transposable elements and the challenges that they present in aging. But obviously we're here at Cold Spring Harbor, the home of Barbara McClintock. So
yes, of course.
So
that's where it all started.
Exactly. So, but for those of us who've kind of forgotten transposons, retrotransposons, and all the differences, can you give us a high-level view of what we're talking about with transposable elements?
Yes, of course. Transposons are essentially genomic parasites. They move within our genomes. They copy and multiply. They come in different forms. Some are RNA-based, some are DNA-based. They have different life cycles. They go back in evolution to the very, very beginning. So all organisms have transposons, bacteria, everything. In fact, they're very conserved evolutionarily. So the element, the retrotransposon line 1 that we are working on is found in plants, bacteria, archaebacteria has obviously been highly successful in our genomes.
Yeah, and you said in your abstract that it's the most common one in humans, is that right?
It is, well, it depends how you define common. In terms of copy number, there's a related transposon, the signs. They're actually more numerous. They use the same mechanism of transposition. What I meant to say is that this is the only transposon in the human genome currently that is active. It's autonomously active, so in fact it can catalyze its own transposition from one site to another.
So how does it move around then?
Well, it starts out in your genome as any gene. That's why they're called jumping genes. It's transcribed by your RNA polymerase II, very obligingly. And then the message encodes some proteins that then bind to the message and then catalyze the entry back into the nucleus and the reverse transcription that enables the retrotrans. So it's exactly the same life cycle as HIV. And actually HIV is a retrotransposon that has acquired or has the ability to move horizontally from cell to cell. So it packages itself into a little viral particle, just like any virus.
Right. But so the lines are just spreading within the same cell.
Exactly. They're kind of stuck in the cell forever. Yes.
And so this is why we have so many. How many are there in the genome?
There's about 500,000 lines in the genome.
Right.
The human genome, about 17% of the human genome is lines, but the vast majority of those copies are ancient and they're inactive because essentially, this particular, parasite, the strategy is to be as benign as possible.
Right.
So they only transpose in germ cells and in early development, right? That's when that's their window of opportunity to be passed on to the next generation.
And why? So what happens during that window of opportunity that allows them to do that?
The genomes become more open and allow their expression.
Right.
Now, of course, it's a evolutionary, warfare. So we know this. Because if we look at their sequences, we can see that they evolve incredibly quickly.
Right.
So the hosts try to mutate them, and then they mutate to escape that survey. It's exactly, the virus paradigm.
So are they at other times are they shut down as heterochromatin, something like that?
Exactly, and that's where this whole aging angle comes from. Because until very recently, it was believed that they do their job, for their survival during that very narrow window of time, and then in our adult bodies, they are essentially completely shut down, they're heterochromatinized, and they're just completely inert.
Yeah.
And what we have found, and many other groups have found as well, is that in fact the surveillance breaks down, and it breaks down with aging. So we think it's actually one of the consequences of the multifactorial failures that happen during aging, the surveillance process becomes more porous, allows these elements to become expressed again, and then they enter into their life cycle, they get into the cytoplasm, and guess what? The cell recognizes them as a virus.
Right.
And it, at this point, it's completely futile for both the host and the parasite. It's futile for the parasite because if it transposes, it's not going to make any difference. It's going to die with you because it's in a somatic cell, right?
Yeah.
And it's futile for the host because, in fact, what the host does is it mounts an antiviral response which results in chronic inflammation. And that, in fact, promotes aging processes.
Right.
So it's kind of a breakdown, which I mean that's what aging is, right?
Yeah, yeah, and of course, I mean chronic inflammation in numerous different tissues is a characteristic of aging. To how many?
Well, that's why we
cases?
got so excited about this, because we realized that this might be an explanation. Probably not for all of this inflammation that you refer to, but maybe some important aspects of this inflammation in diseases like neurodegenerative diseases, cancer, et cetera, et cetera. So there's actually very good evidence that it's the nucleic acids, it's the products of the reverse transcription that are recognized by the innate immune system, and then that triggers these inflammatory responses.
Right, because every cell is conditioned to be able to develop those inflammatory responses. And this is happening in the brain, then?
We think this is happening in the brain, and that's why we're doing now clinical trials on ALS and Alzheimer's disease, because we're repurposing a class of drugs known as reverse transcriptase inhibitors. You're probably familiar with the most famous one, AZT, which was developed for HIV, right? And since the mechanism, the biochemical mechanism, is so conserved, some of these compounds actually inhibit the reverse transcriptase of line 1 elements. And so we've shown this in mice, in cell culture, and there's epidemiological data in humans. Obviously, there are millions of people that are taking these compounds. They're widely used to treat HIV, and it's been shown that there are at least indications that certain diseases such as cancer, Alzheimer's disease, type 2 diabetes, that their incidence is slowed down or decreased in people that are on these therapies. Right,
and that's basically looking at people on the triple cocktail for antiretroviral therapy.
Correct.
Now we have decades of data on that.
Yes.
Yeah. So wow, well that's interesting. I mean, do you think that this means that we're all going to have to go on antiretroviral therapy aged 50?
Well, that is one possible outcome, yes. Yeah. I mean, I think it remains to be seen how broad this principle is and how many diseases it affects. There are actually indications now, exactly as you alluded to, that this might be, in fact, an anti-aging intervention. So, for example, if you look at the DNA methylation clocks, they've received a lot of press. I'm not sure if you buy everything that's said about them, but notwithstanding, reverse transcriptase inhibitors profoundly slow down the epigenetic clocks.
Right.
And this has now been shown in multiple organisms and using multiple clocks.
Yeah.
So from that point of view, aging is slowed down. In model organisms such as nematodes, flies, these compounds extend lifespan. So that's easy to demonstrate in the laboratory.
Yeah, and reasonably well tolerated as well.
They are well tolerated. There are side effects, but there have been successive generations of newer and newer drugs. So AZT obviously did have some serious side effects. It was replaced by other drugs in the same class. And then those drugs have been replaced with even newer drugs. So I think what is really happening now is developing compounds in that class, reverse transcriptase inhibitors, that are now specifically targeted to line 1. because the ones that were developed for HIV, I mean, nobody ever thought of line 1 when those were being developed, right? And so they, they work,
Yeah.
but they're not particularly effective. And so the company I co-founded, Transposon Therapeutics, now has a drug in clinical trial. So this is now an investigational reverse transcriptase inhibitor that's something like 20 times more potent than the generic HIV drugs. And that's now going into a large clinical trial of ALS.
Yeah. Well, it's a fascinating story when we think of the people increasingly comfortable with the idea that in later life you might be taking, I mean, people took half an aspirin a day for years, statins, various things like that. It would be fascinating to see if this drug becomes one of those.
I think statins is actually a very good example of a drug that has had a really profound effect on human longevity. Yes. Yeah.
Well, good luck with the trials.
Thank you.
And,
Thank you very much.
And I hope you have the same success as statins. That would be fantastic. It's been great talking to you, John. Thanks very much.
Thanks for the interview. Thank you.
Okay.